Hello, my name is Autumn Burleid, and today we're going to be discussing the paper Spatial Transcriptomics Reveals Substantial Heterogeneity in Triple-Negative Breast Cancer with Potential Clinical Implications by Wang et al., published in 2024 in Nature Communications. Here we're looking at figure 3A from the paper, and I wanted to start off with this so that I can show you the H&E scans that they took. The stain is used to see the morphology of tissue, and in this case, they're using the scans to group the tumors into categories. I'm currently a histotechnologist, and so I have done immunohistochemistry breast panels that a pathologist will order when they suspect a patient has breast cancer. These panels are typically made up of ER and PR, which are your estrogen and progesterone receptors, and HER2, which is a protein that regulates cell growth and repair. Triple-negative breast cancer means the cancer cells do not have receptors for ER, PR, or HER2, and usually this results in poor prognosis for the patient. Multiomics has been performed on bulk triple-negative breast cancer tumors and created molecular subtypes such as basal-like, immunomodulatory, luminal androgen receptor, mesenchymal, and mesenchymal stem-like. What researchers report from these multiomic studies is that each of these subtypes behave uniquely and have different clinical outcomes. For example, the immunomodulatory subtype is known to have higher expression of immune gene signatures and potentially targetable immune checkpoints, as well as better prognosis. While RNA-seq has been helpful in these regards, RNA-seq does not capture the spatial layout of the tumor, how interactions between genes can affect the phenotype of the tumor or the tumor microenvironment, what types of cells are around, and how are those cells interacting and influencing each other. We can try to answer these questions instead by using spatial transcriptomics. Spatial transcriptomics is a promising new tool because it allows for the investigation of tumor heterogeneity as well as the tumor microenvironment. When somebody has a tumor, the cells in the tumor are not just tumor cells. There's a lot of different types of cells in there, and spatially locating all of them and understanding how they interact with each other could provide a lot of helpful information. And these improvements fill the gaps left by other multi-omics techniques, such as the bulk RNA-seq that we mentioned previously. Figure 7b is showing clusters of cell subtypes within a tumor. Each subtype within the basal-like tumor is represented by a different color. The areas in black, white, red, green, and pink are all different basal-like clusters, while the blue is representing an immunomodulatory cluster. Here we're looking at a graphical abstract that the authors decided to put in here because there's so many different moving parts to this study, so being able to visualize it will definitely help you understand. So in a retrospective study of 92 patients using fresh frozen specimens, the researchers obtained 4 consecutive cryosection slides and collected scrolls for bulk RNA sequencing, which this was already a huge improvement from previous studies where a limited number of patients were involved. 92 is really impressive. So from the 4 slides, 3 were used for H&E staining, and then they were scanned and stripped, and this will allow them to be used for spatial transcriptomics moving forward. The remaining slide received double IHC for CD3 and CD20. From there, morphological annotations and spatial analysis were performed to further characterize the morphology and understand the graphical layout of the stroma, which is the non-tumor cells, as well as the tumor compartments, and then finally clustering. Next, the authors did morphological analyses using spatial transcriptomics global pseudobulk data. And pseudobulk data is basically an approximation of the types of gene expression profiles. When somebody has a tumor, it's not just going to be a tumor. There's going to be other types of cells in there, such as like blood vessels, fats, some areas might be necrotic, there are nerves, the list goes on, but the researchers wanted to understand the molecular features of a tumor and how those features control its classification. As I mentioned before, non-tumor compartments within the tumor are referred to as stroma. And you'll notice that there are a lot of acellular stroma in these tumors, meaning more pixels of stroma than tumor cells. So what could the makeup of these tumors tell us? Here we're looking at figure 3C, and we're looking at the contents of the tumor by subtype. You'll notice that the last 3 right here, mesenchymal, basal-like, and immunomodulatory, they have a bit more green, and the green represents tumor cells. And the luminal androgen receptor and the mesenchymal stem-like have increased levels of the TAN, which correlates to the stroma cells. And basically, the researchers wanted to look at the spatial characterization of these tumors within each subtype. And what the results of this show within the subtypes of the triple-negative breast cancer tumors is that there are these distinct patterns that I just mentioned. And they wanted to know, could these patterns be taught to a deep learning model to determine molecular subtype off of a simple H&E stain, which is obviously much cheaper than doing an entire spatial transcriptomics study on a patient? They analyzed tumor and stroma pseudobulks to give them an approximation of gene expression coming from the tumor and the stroma cells. The results of the spatial deconvolution showed that tumors with a higher number of tumor cells within their microenvironment had increased proliferation and increased immune signaling than tumor cells that were distributed into smaller groups within the tumor. These smaller patches had increased metabolism as well as increased hormone signaling. Classification based on tumor-only compartments revealed 3 subtypes: luminal androgen receptors, mesenchymal, and basal-like, further suggesting that RNA-seq results could rely on stroma features for categorizing immunomodulatory and mesenchymal stem-like subtypes. Hopefully, you are starting to see how spatial transcriptomics could give scientists a greater understanding of triple-negative breast cancer tumor heterogeneity. Both stroma and tumor compartments are critical for the classification of triple-negative breast cancer, and spatial transcriptomics reveals what RNA-seq simply cannot: the heterogeneity of tumor cells. Findings for stroma compartments from supplementary tables 10 and 11 find that mesenchymal stem-like stroma have increased angiogenesis, so the increased formation of new blood vessels, and increased cancer-associated fibroblast signals. Mesenchymal stroma were found to have increased neutrophil infiltration and lower immune signals. Luminal androgen receptor stroma were found to have increased adipogenesis, so formation of adipose. Immunomodulatory stroma had increase of multiple immune signals, and an interesting clinical finding of that is that patients who had a mesenchymal subtype were more likely to remain in remission when the stroma was mesenchymal stem-like and had increased immune signaling compared to having stroma that was just mesenchymal. So that's an interesting clinical finding. Earlier I mentioned that 3 consecutive cryosections were taken. And that's really important here because they use that for imaging to build the spatial transcriptomics analysis off of. So when somebody is cryosectioning, usually they have their piece of tissue and they're going in from anywhere from 4 to 8 microns at a time. So with 3 consecutive sections, most likely you're getting a pretty good range of space within that tissue. So the first is usually the most superficial, and then you go in a little bit more, and then your third one's going to be the deepest one. And so what they were able to do was better understand the spatial relations between this tumor when they had 3 consecutive slides to look at and then annotate and then move forward with imaging. So, identification of spatial molecular patterns using clustering. What they found was that across 92 patients, there were 418 separate clusters. Groups of cells sharing similar gene patterns of expression were found, and each patient had between 2 to 8 different clusters per tumor. The analysis of interpatient clustering led to the classification of 14 separate megaclusters that are commonly found across interpatient clustering. Of those 4 megaclusters, 9 patient-based spatial archetypes were noted. Through their research showing the different contributions to the molecular makeup of these tumors, they were able to determine several characteristics and potential therapeutic targets. They discovered that the immunomodulatory subtype is composed of mostly basal-like tumor cells that have high levels of lymphocytic infiltration.
as well as that the mesenchymal stem-like subtype was associated with stroma-specific characteristics, and the mesenchymal subtype associated with mesenchymal in the stroma was related to less favorable outcomes compared to mesenchymal with mesenchymal stem-like in the stroma. These findings reiterate the hypothesis that spatial transcriptomics is highly relevant to clinical outcomes for diagnosis and treatment. With this information, simple, routine H&E slides could potentially be used to determine the molecular makeup of triple-negative breast cancer tumors and correctly identify next steps. This could mean better treatment options for those affected by this aggressive form of breast cancer. We also have specific therapeutic perspectives listed in this figure, depending on the subtype, which could be really important for clinicians in order to better treat their patients. There is a bit of room for improvement in this study, especially since it is a really rapidly advancing field. This paper did only come out a year ago, but a lot of the technology is now much more advanced, such as for the image resolution. The spots they analyzed, such as individual bulks, only went up to 200 cells. So this limits the accuracy of the actual tumor microenvironment and the tumor heterogeneity, which are really important factors in this study. Constrained analysis of small structures, such as the tertiary lymphoid structures, also leaves room for improvement. So again, application of more up-to-date technology could improve the study. And the major limitation to this study is cost. I can't speak to how much the author spent on this study specifically, but I used to do geomics, which are a type of spatial transcriptomics, and it could run a PI anywhere from $20,000 to $60,000 for just a couple slides. So for 92 patients at 4 slides each, I would imagine the study cost millions of dollars. IHC immunohistochemistry is still a valid and less expensive replacement for spatial transcriptomics, but it is not as detailed. Finally, the deconvolution method did have some difficulties determining cell types. To wrap things up, this paper showed that spatial transcriptomics of triple-negative breast cancer, in coordination with multiomics, allowed for the categorization of tumor spatial heterogeneity, which shows promising ability to determine targeted treatments. Classification of 14 spatial patterns were exhibited by patients in the study, from which 9 spatial archetypes were depicted using available bulk RNA-seq data. Patients showed 1 to 6 different megaclusters within a single tumor. So there's not just tumor cells in these tumors. Understanding tumor heterogeneity of a patient's tumor could have strong implications for future medical interventions. And these are my references. Thank you for watching my video.